with other people, you know, one of the challenges for folks is sorting through all of the noise that you see in the news, and there's quite a bit of noise. In fact, I would say most of it is noise, and so it can be a bit of a challenge to sort of track it all and what's going on. So kind of today, I thought I'd just visit with you about you know, where these viruses come from that start to invade our lives, and how really our immune responses, our immune systems deal with that under normal conditions. And then just kind of what to expect out of the vaccine efforts that are underway right now. So can everybody see the screen share that I put up? So far, so good? Okay, good. So the nice thing is I'm all set up for this. I've got my whole teaching platform here. So the picture up on the screen now, the one on the left, is one that you've seen quite a bit of. That's our sort of ubiquitous picture of the coronavirus for SARS-CoV-2. And then the cartoon on the right is really the diagram that you'll see when you're trying to figure out how this thing really works. So these little red things on the outside, these are the thing called the spike protein. And whenever you see a picture and you see all these little nubs that are all around the outside, that's that spike protein, and that's the thing that sticks onto your cells. So that's the thing that sticks onto the cells and sort of helps the virus get into the cells and start replicating. The rest of this is most of the other cell biology stuff because it comes off of your own cells, so it's taking bits of it with it. So the membrane that actually wraps around the virus, that's your own cell membranes that have been co-opted to make this sort of packaging material. And then these blue zigzags inside, that's the genome of the virus. So the virus is an RNA virus, meaning that rather than having a DNA genome like we have, it has an RNA genome, which is normally sort of the messenger that goes between our DNA and the machinery that makes proteins. So it actually has this genetic material as RNA. It's not really alive because it doesn't do metabolism. So it's just basically a little set of instructions that get produced in the cell, released, and then stick to another cell and copy itself. So they're kind of creepy in a way because they exist simply because they can exist and not toward any purpose. So that's what the virus looks like. It's called SARS-CoV-2 because if you remember back to 2002, 2003, there was the initial outbreak of something called SARS, severe acute respiratory syndrome, and that was identified in China. And just kind of an interesting timeline when that was first identified is that it was the first patient was found in, I think, I don't have my glasses on, gotta put my glasses on. First patient was found in 2002, in November of 2002. And within about 6 months or so, the disease had kind of run its course and they were able to contain it. So it was a pretty nasty infection. So I think the case fatality rate was in the teens or 20s. But it didn't spread quite so easily. And so in the end, I think there was only 764 confirmed deaths as a result of SARS, even though it was splashed all over the news. And you remember people wearing masks and doing this kind of thing to contain it. They were able to contain it pretty quickly. If you compare it to the outbreak that we're seeing right now, this little section here amounting to the first 4 months of this outbreak, or of the SARS side, this is all that has happened just in the first couple of months of the SARS-2 or COVID-19 outbreak. And really, it just sort of is an example of how quickly this particular virus is moving, because within, 1, 2, 3 months of the first patient in Wuhan, China, we had already exceeded the death toll of the first SARS outbreak, and the virus was global. So this is a slightly different take on the original virus. It's very much related to it, but it's changed in about 15% of places, and that's made it much, much more contagious. So that's why it was a pandemic instead of a localized outbreak, because it's a very good spreader. So I tell people this isn't like the flu. This is a cold that can kill people. So it spreads like a cold and moves really, really fast. But the sort of question a lot of people have is, you know, how do new viruses emerge like this, whether it's Ebola virus or other things like that? And the basic answer is that they aren't really new viruses. So what we have is that all multicellular things, or, you know, in this case, all animals and many plants have viruses that are in them. And what ends up happening is that these viruses circulate in those animal populations, and then once in a while, one will take on the characteristics that allow it to jump from one species to another species and become now a different virus in that species. So, so something will change, and then it'll move, and there'll be the opportunity for spread, and it'll move between animals, and that's how these things emerge in the human population. So, a process we call zoonotic infections. And this paper here that came out was one that sort of gave me shivers because I hadn't seen it before this year. And actually, the first author on this paper is a friend of mine from grad school, Vinny Manicherry. And this paper was from 2016, and it said SARS-like WIV1 coronavirus poised for human emergence. The W is Wuhan, and it was a family of viruses in bats that they knew about as early as, well, even before 2016, where many people have been studying these viruses and seeing that there were many different kinds of coronaviruses in bats that were very much related to SARS. And what got their attention in this paper was that this particular virus that they were looking at was able to bind the same human molecule that SARS uses in order to invade our cells, this ACE2 receptor. And so that was alarming that they'd found multiple different viruses that could now potentially go from bats to humans and cause an outbreak. A couple of years later, actually, in the middle of last year, another group that was studying these viruses found that actually there were people in China who had antibodies against SARS-like coronaviruses in their systems. So basically what that means is that as in the middle of last year, people were being exposed to viruses that are like the one we see today, where it had sort of emerged into the population, but then not really taken off and died off. So it's really kind of interesting in that these viruses that were in bats of this family of coronaviruses, they were jumping from bats to humans multiple times, and it never really took off. They never really were of the right variety to spread. And then it appears sometime late last year, one of them that did have the characteristics to make it spread very easily from person to person, made that transition again and got into the population and started spreading. So when you talk about, or when you hear people talking about the research on that they think this came from bats, it's really pretty good because they've been following these viruses in bats now for a few years, and this one looks just like those. Genetically. So when I talk about looks just like it, it's a matter of taking out that RNA genome and getting the sequence and comparing it, how close it is to these other ones. And it's pretty good now that we can see that these viruses are there in that population and they're circulating. And as early as last year, you had some people who had some responsiveness to those. So that's kind of the history of before we had COVID-19, there were things circulating around in the bat populations in Asia that occasionally jumped into the human population and caused disease and were squashed out. It wasn't until late last year that one spread. So this is a sort of how that virus gets in and spreads to us, and you heard about the aerosolization of it, where it'll come and infect your airways. And most people are first infected in the nasopharyngeal area. So basically it becomes an upper respiratory cold first. And you might get stuffy nose, you might get a headache, you might get all those normal cold-like symptoms. But then as the virus is replicating up here, it sort of starts working its way down and down into the lungs. And at that point, that's when you get the sort of pneumonia characteristics of it. So many people carry it up here as either an asymptomatic infection, or they carry it as something that just feels like a minor cold or an allergy, and then it creeps down into the lungs and causes pneumonia. So some people, it stays up here and never gets into the lungs, and some people get all the way down and have a nasty infection from it. So it's sort of life cycle is, once you breathe it in, it's going to stick onto one of the cells up in your nose in the upper respiratory tract. And that virus membrane that I talked about is going to fuse with the cell membrane, and that's what allows it to get into the cell. And then it just uses your normal cellular machinery to replicate itself. So all the instructions it needs for replication are in that little strip of RNA inside. And then it starts making more copies and making more copies and eventually releasing those copies out so that other cells nearby can be infected. So that's kind of what goes on during the infection. The good news for most people is that we are not totally helpless at this stage. So even though the immune system hasn't really been alerted, you have what we call cell intrinsic defenses that actually detect that there's a viral infection. So every one of your cells in your body has little, what we call sensors or pattern recognition receptors that can detect when they've been invaded by a bacteria or a virus. And we have names for these are RIG1 and MDA5, and you don't really have to think too hard about those, but they sense when there's a RNA molecule that isn't one of your own because the structure is a little different. And they send a signal down to the cell and tell the cell to start sending out an alarm signal. And that alarm signal is these little red dots here that we call type 1 interferons. And they got their name because of the ability to interfere with virus replication. So that was the first people that discovered those, found if you put these in a dish or cells that made these in a dish, they could interfere with the virus's ability to infect surrounding cells. So a cell, when it gets infected, starts making a whole bunch of these things, and it tells that cell and all the neighboring cells to start shutting down the machinery that a virus might use to infect. And so you get a little bit of a slowing down of the process to sort of give your immune system time to get going when you get infected. You also have a set of cells that are called natural killer cells. It sounds like kind of a nasty name for a cell, but they were named originally because they were discovered that they had the ability to kill off some tumor cells. So these are white blood cells circulating in your blood, and some cancers could kill them off, or they could kill off some types of cancer. Well, they also run around looking for cells that look like they're sick, that are giving off signals to say something is wrong in here, something is stressing me out, and the natural killer cell will come in and kill these things off. So, so you have some mechanisms to start fighting the virus right away. One of the troubles as people get older is this system here, this interferon system, to release these sort of signals out here that are signaling danger, and that are also used to get the NK cells going. As you get older, your interferon responses are not as robust. So it's just, you don't produce as much of the interferon system as when you're younger. So that could be one of the reasons why older people suffer from this virus quite a bit more than younger people, because younger people's interferon system kicks off really high right away. And for older people, it's a much more muted process early on. So that's the early things. That's what's happening within a first few hours really of being infected. After a day or so, and around 4 days, the cells of your white blood cells that we call your adaptive immune cells kind of take over the process. And these are ones like B cells, and the job of the B cell is to make antibodies. So you've probably heard the word antibody thrown around on the news quite a bit. And all an antibody is, it's a little protein that's floating around in your blood, and it, if it's recognized that virus before, if this B cell is able to make an antibody that recognizes that virus, this B cell will grow and make many copies of itself and start to make many, many antibodies and release those into circulation. The trouble with this system, though, is you have billions of these in your body, but every B cell can only recognize one shape. So it only has the ability to find one particular molecular shape anywhere in the world. So what happens is you end up waiting for the virus and the B cells to circulate around enough to the point where the virus runs into a B cell that can recognize it, and then that B cell is activated and grows and grows and grows and starts making the antibodies that would neutralize the virus. So this process takes a few days, and really takes about a week to get going. So that's why when you have a cold, a lot of times you are going to be sick for at least a week, because it takes the cells that would make the antibodies at least a week to get going and make enough antibody to sort of neutralize this. You also have another class of cells called T cells. And if you're familiar with HIV AIDS, T cells are the ones that once they are depleted, so HIV infects T cells, and then the T cells get killed off. And once all those T cells are gone, that's when you get AIDS. And the job of the T cells are to, some of them help these B cells to grow and divide, and some of them actually kill infected cells. And so these are the cells that make up what we call your adaptive immune system that takes many days to get going along the way. So we have what we call the helper T cell. That's the one that I mentioned HIV gets involved in. And then the cytotoxic T cell is the one that actually goes around now and kills infected cells in your body. Once you've got these going, now you can actually clear an infection. So there's a lot going on in the immune system in the first week that you get infected with something. What these also do that make things work better for you later on is they form what are called memory cells. So I talked about you might only have one or a few cells that recognize the virus when you first get infected. When you actually go through the whole immune response, you make thousands or millions of those. And so next time you get infected, you've got thousands or millions of those that are ready to respond, and it doesn't take you much time to respond anymore. So things are much better and much faster. So SARS-CoV-2 or COVID does the same sort of thing, that once it replicates, it's released from the cells, and some of the virus goes on to infect other cells, but some of it is picked up by your immune system, and your immune system uses those pieces to actually mount an immune response and say, okay, this is something dangerous and I'm going to fight it off. So I like to show this curve, and actually this is one that is in chapter 1 of just about every immunology textbook ever read. And what this is showing on this left axis here is the antibody titer. So if you were to go into the clinic and they were to look for coronavirus antibodies for you, and maybe some of you had this test already, what they're looking for is in the blood test is to try to see if you've developed antibodies to the coronavirus. And when you first get infected by something, you have what's called a primary response. The antibody levels go up for a few weeks in the serum, and basically once you're getting to this point on the curve, now you've got enough antibody there to clear the infection. So essentially by this time, if you've got this antibody response, you're not going to find virus in there anymore. But what you notice is that response tapers off over time. So you might have heard a lot of news reports of people sort of becoming hysterical because the antibodies to COVID were going away after a few weeks or months after people got better from infection. And actually that is entirely normal. That's not just a coronavirus thing. That is every single infection you'll ever get will follow that same timeline of you have an initial response to clear the infection, and then you don't really need to keep those antibodies around anymore. So they taper off and you're not going to make them. But what happens is some time will pass and those will disappear. But if you get sick again, if you get reinfected by the virus, now you're going to have a much stronger and much faster second response. And that much stronger and faster second response means you don't get sick from the same thing more than once, or actually not real sick. So the question of whether you could be reinfected by COVID, sure, you could be reinfected. But the second time you're infected, probably you're going to mount a much faster immune response and you're going to have a much milder infection as it goes on. And then once that starts to taper off too in the end, after you've been exposed multiple times, you'll sort of maintain a higher level antibody response than you would have before. So that's why whenever you get vaccinated, what you'll notice is that most vaccines, you're given a shot and then you wait some time and you're given a booster shot. And the whole point of that booster shot is to go through these phases in order to protect you so that you've already done this before getting exposed to an illness. So really when the vaccines come out, what we're trying to do in the first shot is get this process over with so that if you get infected after that, you can go straight into the secondary response and hopefully not have a very severe infection. And if we get booster shots, maybe you can go through both of these phases and you'll be protected from infection later on. So that's kind of the goal of any vaccine program. But when you hear them sort of talking about, that our immune responses are waning, your immune responses aren't really waning. It's part of the normal process of an immune response over time. So it's one of those that as it's been talked about, I haven't really even worried about it. So really what we're trying to do with the vaccine programs is get you through these first phases, those first humps, as it were, to get your first responses underway, and then you can be protected. So you probably heard in the news about the different virus platforms out there. One of them It's been reported on a lot as one from Oxford University that's being manufactured by AstraZeneca. This one is actually based on putting the gene that encodes that spike protein into a different virus vector, what's called an adenovirus. And it's another type of virus that causes colds. And they actually took this cold virus from a chimpanzee, put the gene for SARS spike protein or COVID spike protein into that, and then stick that in your arm to infect your cells. And what that does is cause an immune response against the spike protein to help block the coronavirus infection. There's another one or two out there that are also leading contenders from Pfizer and Moderna. And they don't use a virus to infect. They use almost like a little sac, little fatty lipid sac around this RNA to make the spike protein. And then you stick that in your arm to have your cells make that spike protein, and then your immune system will recognize it as foreign and start to make antibodies to that. So these are the two or the three sort of leading contenders in the vaccine race right now that are ahead of the pack. If you've been watching the news in the last day or so, you'll have found out that this vaccine trial has been put on hold because somebody developed, they haven't confirmed this, but it sounds like a central nervous system inflammation. So they're waiting to see if that inflammation was a result of the vaccine or just coincidental. And so that they've stopped recruiting now for this one and they're on hold. And the Pfizer and Moderna vaccines trials are still underway. The trouble with all of these systems is that actually there aren't any approved vaccines right now that are in these formats that are in sort of the first wave of trials. So when you hear about vaccine trials reporting by the end of the year, we'll be watching for that data, but those are ones that are all experimental and haven't been really trialed in humans before. If you're familiar with other vaccines, so MMR vaccines, polio, smallpox, influenza, those many times use attenuated virus. So if you've gotten the seasonal flu shot in recent years, it's been one where the virus has been changed to somehow make it not very good at replicating in people, but you still get a good immune response. There's another type of influenza virus where they just simply grow the live virus, but then inactivate it with formaldehyde, the same thing used in embalming fluid. So they chemically inactivate it. And then there's another type of a replicating one, and the Ebola vaccine is something like this. And then there's another way you can do it of just using a protein, like say they're using the spike protein. The tetanus vaccine uses this sort of strategy. So there's a lot of different strategies out there for making vaccines, and they're all in play right now, which is good. But the tricky part about it is when they talk about that they're in phase 3 trial. The phase 3 trial is the really important one because that's the one where they include tens of thousands of people and they check to see whether it works to prevent the illness. And they also check to see, importantly, is it safe, because you're about to inject this into billions of healthy people. So obviously you don't want something dangerous. So that's kind of where we are right now on the vaccine trials. And I'll sort of skip through this because I talked about it, about how these different vaccines really are working. But just kind of when we think about expectations, there's been a lot of hype on the news about one of these coming out very soon. If we go back and look in recent history, it was 15 years from the time Ebola was discovered till we had a vaccine approved. More recently, for the most related coronaviruses, SARS and MERS, after 6 years or 17 years respectively, we don't have approved vaccines for those two illnesses yet. And part of that is because they were very limited in their scope, so the sort of global push to develop them wasn't that big. But right now, when they're talking about Operation Warp Speed, we're talking about taking a vaccine, taking a virus that we've just got the sequence of early in the year and getting a vaccine within 12 to 18 months. And that would be beating the old record by approximately 3 years. So the fastest vaccine ever been made and approved was 4 years, and we're trying to do it in under 1. So that's when people say, oh, it's imminent or this or that. It's like, well, be patient, because you want the safety trials to to get done and know that you've got something worth having, and not pushing that out too soon. But that's kind of where we are on the vaccine process. I would say, the take-home points from all this is that, COVID has the potential to be a very nasty illness for some people. And by and large, the older you are, the more dangerous it is. But as far as how your immune system is responding, it's actually pretty textbook. So it's your immune system is responding just exactly how we would expect it to any new virus infection, and it's not so unusual there. And so that's actually really good news, because when we talk about trying to make a vaccine for it, we kind of know what we're doing, although we're pushing our timelines a lot more than we used to. So that's kind of where I'll end my story on this today, and I'm happy to take any questions that you might have.